Hi everyone. So in the first two sections of Chapter 1, it's been an overview of genetics. Section 1.1 was largely focusing on sort of what the genetic material is and how it works at the molecular level. So DNA is organized into units called genes that are expressed such that RNA is made, and then the RNA encodes polypeptides that constitute proteins. Those largely are responsible for cellular structure and function. And then in the next section, 1.2, we kind of took that to the next levels, which involve thinking about how genes affect the traits of multicellular organisms, how those genes are passed from parent to offspring, thinking about sets of chromosomes, that sort of thing, and the idea that genes are passed from parent to offspring. And then we even thought about genetics a little bit at the population level in terms of our butterfly example, where there is different genetic variation, and the lightly colored butterflies tend to be out were more prevalent in light meadows, whereas the darker ones were more prevalent in forest. So it was just kind of an overview of how genetic works at different levels. In section 1.3, we're going to begin by talking about model organisms, which are particularly important in the study of molecular and organism transmission genetics, as we'll define in a few minutes, and then talk about the three fields of genetics. So researchers oftentimes study model organisms so they can compare their research results. So E. coli is a bacterium. It's been really important in terms of learning molecular genetics. Saccharomyces cerevisiae is also a microorganism, but it's eukaryotic, not prokaryotic. It's also been very important for understanding eukaryotic molecular genetics. Drosophila and C. elegans have been important for understanding somewhat cellular genetics, but especially how traits are passed from parent to offspring and how multicellular organisms develop from a fertilized egg. The same is true of Arabidopsis. The mouse and Arabidopsis are multicellular organisms that people tend to care a lot about. So a mouse is a mammal, and a lot of the characteristics of mice are similar to what you'd find in humans, which are also mammals. Arabidopsis is a flowering plant, and so a lot of the crop species of plants are flowering plants. And so there's a lot of interest in them from that perspective. Genetics is traditionally divided into three fields. There's transmission genetics, which follows the transmission of traits from parent to offspring. There's molecular genetics, which is thinking about genes at the level of DNA and the synthesis of RNA and proteins. And there's population genetics, which is sort of at the other end of the spectrum, which is thinking about individuals and populations, usually out in nature, what type of genetic variation it contains and how the genetic variation changes from generation to generation. So as I just mentioned, transmission genetics explores inheritance patterns. It's the oldest field of genetics. It's where Gregor Mendel started his famous studies. It basically examines how traits are passed from one generation to the next. In Chapter 2, we'll go through Mendel's work, which is historic. And he identified these units, these discrete units, which we now call genes. In transmission genetics, the basic experimental approach is a genetic cross. So two selected individuals are bred to each other. This doesn't apply to humans. In humans, we do something different called a pedigree analysis. We'll talk about that in Chapter 2. And then the traits in question are analyzed over several generations. The analysis is typically quantitative in nature. And so that's transmission genetics. And the first 8 or so chapters in your textbook are focused largely on transmission genetics. The biggest bulk of your textbook is focused on molecular genetics, and so that's from chapters 9 till the last 3 or 4 chapters in the textbook. It focuses on a biochemical understanding of the hereditary material. And so we largely think about things at the level of DNA-based sequences in genes and RNA molecules and proteins. A lot of it is about how genes are organized and how they're expressed at the molecular level. And it tends to involve a detailed analysis of DNA, RNA, and proteins. And they typically employ a genetic approach, and what that means is a lot of times in molecular genetics, what researchers do is they identify mutants that don't function properly, and they compare the mutant with the non-mutant organism, and that helps you oftentimes to figure out what the functional or non-mutant gene actually does. By eliminating its function, sometimes it gives you a strong clue as to what the non-mutant gene does. For example, if you knock out a gene that's involved in blood clotting, then the blood doesn't clot properly, so that gives you a clue that the functional gene plays a role in blood clotting. Finally, the third field is population genetics. It's concerned with genetic variation and its role in evolution. It largely deals with genetic variation, as we talked about in the butterfly example in section 1.2, and how that genetic variation is related to the environment. You know, why are certain traits found in certain environments rather than others? And what population geneticists do is they develop mathematical theories to explain why certain alleles are prevalent in populations. Now, one of the things that I do is I occasionally tell jokes. Oh, yes, I do. And they're all genetics jokes. And they're all like, what do you get when you cross jokes? Okay. So here goes. What do you get when you cross a fawn, like a fawn is like a baby deer, and a ghost? What do you get when you cross a fawn and a ghost? You can pause if you want to think about it. You get bamboo. Bamboo. That's so bad. It's so bad. You see, a fawn is like Bambi, but a ghost says boo, so you get bamboo. Okay, never mind. All right, never mind. Okay, in the next section, we're going to talk about a bit about experimentation and the science of genetics.